Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 23rd November 2020. And as all of us know, we are into the revision mode. We are revising all important chapter number 12, biotechnology and its applications. As I have been telling to you time and again that these chapters of biotechnology are not just for the sake of CBSE or NEET exam. These are very important as far as your career is concerned. You can as well become a biotechnologist, and biotechnology is the future of the world and the human beings. Hence, biotechnology need to be given a lot of importance both in our country as well as in the world, because this solves many problems naturally that human beings are facing today. Hence, we need to focus on that. So we were talking in our previous session biotechnological applications in medicines, and we talked a very important application in preparation of human insulin. Thanks to the company Eli Lilly for that, and thanks to two scientists, Bantic and Best, along with of course McLeod, who have started off this initiation of preparing insulin from pancreatic islets of a dog, and that is the starting point of thinking. So thanks to the thinking of these two scientists. So we move ahead with another application called gene therapy. So it is a collection of methods that allows correction of genes. This is important. Correction of gene defects that has been diagnosed in a child or embryo. This method is applied in a person with hereditary diseases. See, what is our belief so far is hereditary diseases cannot be cured. Now we have got a ray of hope through biotechnology, through the applications of biotechnology in medicine, in the field of medicine. Even incurable hereditary diseases can be cured. That is the greatness of this biotechnology. In this method, genes are inserted into a person's cells and tissues to treat a disease. This is gene therapy. The correction of gene defect involves delivery of normal gene into the individual or embryo to take over the function and compensate for non-functional gene. So this is step number 1. We are going to introduce a normal healthy gene in place of a non-functional defective gene. The first clinical gene therapy was done in 1990 to a 4-year-old girl with ADA deficiency. What is this ADA deficiency? A for adenosine. You remember adenosine, nitrogenous base. Deaminase deficiency. Adenosine deaminase deficiency. This disorder is caused due to the deletion of gene. See, gene is deleted for adenosine deaminase. That is essential for immune system to function. If immune system is our defense system, all the soldiers will be present there. If immune system is weak, means our soldiers are weak. If our soldiers are weak, who will protect us? That is the question. So this defect can be treated by enzyme replacement therapy. Enzyme replacement therapy, in which functional ADA is given to the patient by injection or bone marrow transplant. In gene therapy method, lymphocytes from the blood of the patient are grown in culture medium outside the body. A functional ADA cDNA is then introduced into these lymphocytes and returned to the patient. So what we are doing, first we have to take the lymphocytes from the blood of the patient, grown in culture medium outside the body. Then functional ADA cDNA is then introduced into these lymphocytes and returned to the patient. So in this method, periodic infusion of such genetically engineered, this is important, genetically engineered lymphocytes is needed. If gene is isolated from bone marrow cells, producing ADA is introduced into the cells at early embryonic stages, it could be the permanent cure. Understood? So we have got two ways. One is the enzyme replacement therapy and another is the lymphocytes with ADA cDNA. But every time the patient is to be given infusions. So instead of that, what we are talking is, instead of introducing cells, introduce gene only. First isolate the gene, and if the gene isolated from bone marrow cells producing ADA, that is adenosine deaminase deficiency, is introduced into the cell at early embryonic, condition is there, it should be early embryonic stages. That could be a permanent cure. Look at this, this is what we are doing here. So plasmid is there and the DNA is identified. So gene of interest incorporated into this, and the lymphocytes are prepared and inserted into the baby or embryo. So this is what we do through gene therapy. So basically this adenosine deaminase deficiency, this enzyme is very important for the immune system to function. SCID is caused due to defect in the gene for the enzyme adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation, as we said. And in others, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. But in both approaches, the patients are not completely cured. This is the defect. However, if the isolated gene from bone marrow cells producing ADA is introduced into the cells at the early embryonic stages, that can be a permanent cure. In case of many pathogens, the gene encoding a critical antigen has been isolated and expressed in bacteria or animals, and the recombinant protein so produced is used as a vaccine. Such vaccines are called recombinant vaccines. A constituent polypeptide in case of hetero-multimeric proteins or a small part of polypeptide may be used as a vaccine. Such vaccines are known as subunit vaccines. And of course, monoclonal antibody is a specific antibody produced from a normally short-lived antigen-activated B cell that has been immortalized by hybridizing it with myeloma cells by hybrid cell culture technique known as hybridoma technology. This is also done in that. So we also have that molecular diagnosis, a conventional method of diagnosis such as serum culture, urine analysis, is not able to help us in early detection of disease causing pathogens or virus. Following methods can be used to diagnose it early. What is that? 1. Recombinant DNA technology or DNA technology, which we have been talking. PCR, polymerase chain reaction. Enzyme linked immunosorbent assay, ELISA. Now this ELISA is frequently asked in CBSE exam for expand ELISA. One more question. Enzyme linked immunosorbent assay. Symptoms of diseases appear only when the concentration of pathogen gets increased significantly. Low concentration of bacteria and virus can be detected by amplification of nucleic acid by PCR, polymerase chain reaction. It detects the mutation in the gene in cancer patient. PCR is routinely used to detect the HIV in suspected AIDS patients. Genetic disorder can also be detected by using PCR technique. A single-stranded DNA or RNA having radioactive molecule is allowed to hybridize to its complementary DNA in clone of cells, followed by detection using autoradiography. The clone having the mutated gene will not appear on the photographic film. So ELISA is based on the principle of antigen-antibody interaction. Infection by pathogen can be detected by the presence of antigens like proteins, glycoproteins, or by detecting the antibodies synthesized against the pathogen. And same thing, we are also, scientists are talking about the corona also, plasma test. What is that they are saying today? The antibodies that are being produced in that. So many tests are helpful in curing the once thought of incurable. So that is the strength of biotechnology. So in our next class, we talk about the transgenic animals, and then is there any ethical issues involved in that? Is there any ethics to be followed when we perform the experiments in biotechnology? And will there be a bio war in future when the ethics are kept aside? Let us reflect upon that in our next session. Until then,